monthly meeting of the Mycological Association of Washington, D.C. I am the president, Elizabeth Hargrave, and I'm going to spend just a few minutes going through some club news and some fungi in the news stories. And then I'm going to hand it over to April Thompson, who's going to talk about culinary things that have been happening, especially in the last few days. Annie Weissman's going to talk about some of the mushrooms we found at our Sequinoda retreat. And then our main talk is Gabrielle Sorbeville, the chaotic forager. And first off, I just want to share some pictures from our amazing event that we just had over Labor Day weekend out at Sequinoda Camp and Conference Center in the Laurel Highlands of Pennsylvania. We found a great mix of mushrooms. We had 4 guest mycologists. This is Bill Ewell, Alan and Arlene Bissett, and Kurt Miller, all the way up from Puerto Rico, who were helping us out. Beautiful woods up there. It was a little drier than it's been the last couple of years we were up there, but there was still plenty to be found. And we had a bunch of other workshops as well. So someone did paper making with mushrooms, some false turkey tail, and a few other species. We did a mushroom jewelry workshop with Saranella, casting things in resin. There was lots of cooking of mushrooms to be done and delicious eating. We had a talent show with mushroom songs and costumes. And just a great spread of mushrooms that our expert identifiers put names on so that we could all learn a little bit more about how to ID things. If you were there, or if you weren't, some of these links may be of interest to you as well. We've got a feedback form for the folks that were at Sequinoda, and please do fill that out. We have a Google photo album with many more pictures than what I just shared, and folks are welcome to continue uploading photos there as you get to them. And then Annie in a little bit is going to talk more about the iNaturalist project that we started specifically to capture just the pictures from this weekend, and it's pretty cool. And stay tuned for that in a few minutes. Oh, one of the charts that's on here is not showing up, but oh, there it is. Great. There are 2 more forays that I just wanted to point out this year. There's also a NAMA foray, but that one's sold out. But in a couple weeks, there's a link-off foray up in Pennsylvania again, around Pittsburgh. It's a shorter event, but has a very good reputation as finding good stuff and good people. And then the Northeast, I believe it's Mycological Federation, NEMF, has a weekend foray up on Cape Cod, and that registration is still open until September 6th. It looks like they've got a few slots still available if anyone wants to make a last-minute trip up to Cape Cod this fall. And then do save the date for our next trip up to Sequinoda, which will be Labor Day weekend of 2025. All right. Lots more events going on this month. Mount Rainier Nature Center is hosting a whole series of things because September is Mushroom Month. So first up is what Saranella says is her favorite mushroom movie, a documentary about fungi. So she's going to show that and then do a panel afterwards with Tom McCoy and herself. Our regular monthly DNA day is at Mount Rainier Nature Center normally, so they've worked that into Mushroom Month events. So you can take stuff in the morning to be sequenced, or you can just go ask questions in the afternoon. There's an arts and crafts workshop on September 12th. We're doing a launch party for my new mushroom themed game called Undergrove on September 19th. It'll actually be out in retail in October, but if folks want to come to that, I will have a bunch of copies to use to teach people how to play. And then a night hike, which we also had one at Sequoia. There's so much to see that will fluoresce in ultraviolet light, and there are a few bioluminescent mushrooms in our area that should be out this time of year. So that should be a fun one too. All of these, some of these have event listings in the PG County Parks system, but you can get to all of them from our website, from the calendar that's on the front page. We made listings for them there as well. Other stuff, our monthly program is always the first Tuesday of the month. And then one of our other biggest events of the year is a mushroom fair that we have at Brookside Gardens every year up here in Montgomery County. Um, and that's going to be October 13th. We will probably schedule a bunch of forays ahead of time. And folks are welcome to also bring in mushrooms that you find in your yard or in your neighborhood or wherever to bring in, and we'll ID those and we'll get them set up on the table for everyone who comes through Brookside Gardens that day to see. We usually get a lot of just random passersby, and then a lot of people come out from the club. It's a great event. April has just scheduled a mushroom beer making workshop for October 27th, so stay tuned for that. You'll get a separate email on that one. And then two deadlines I wanted to point out. MAW offers research grants to folks who want to do academic research projects focused on mycology. If you know someone who might be interested, please pass this along. The info is under scholarships on our website, and we'll give up to $1,500. And then we have the folks who get a grant come do either a full length or a shorter presentation in one of our monthly meetings. So it's a win-win. And then Tom McCoy's running a photo contest again this year, and wanted to remind everyone that that deadline is November 11th. So he and his team can pick their favorite photos by the end of the year. So lots going on. It's a great time of year for mushrooms too. So I'm sure there will be forays scheduled among all these dates as well. All right, I have just a few fungi in the news stories. The first one, there was a paper that just came out describing two new species of psilocybe in southern Africa. One was in Lesotho and one was in South Africa. They were both species that were first sent to mycologists for ID by amateur mushroomers, which is fun. So there's sort of a community science aspect to both of these. Both of these the Latin names that they gave them are references to local mountain ranges where they are found. And so, people often call these mushrooms were just discovered, right? But they weren't discovered, it turns out, because the team actually went to the trouble of going and doing some interviews in the region about whether anyone already knew about these mushrooms. And sure enough, there is a history of use in that region of Africa by local healers and shamans, where they actually mix it with a hallucinogenic plant and give it to people. And then they have sort of a guided trip where they're sitting with that healer who's helping to interpret what their visions are. So I thought this was a really interesting case of a paper where they went and sort of did the ethnological, anthropological research along with just describing the new species. Is a nice example of that. All right, I'm going to try and play this short little video from NASA about what they're doing with mycelium for possible architecture on Mars and the moon. Is this going to play? So as far as I can tell, this is like a internal grant program that it's actually NASA staff who are working on growing th structures made out of mycelium. But they've put out a press release about the fact that they've awarded this project. So that was kind of fun to see. I know we've had several items in the news lately about building with mycelium, and it seems like it's catching on. Oops, I don't want to play that again. I want to go to my next slide. Here we go. I have another video for you about this project that is using fungi as part of robots. So did everyone see the flashlight that just came down and shined UV light on this? There is mycelium in this robot that is responsive to the UV light. I'll play it again. Oops, it went big. Look at that. So the UV light, and then it's like picking up on the electrical signals that the mycelium is making in response to that light, and the robot goes faster. So that seems cool. And I think that is all I have for the news. So I'm gonna stop sharing my screen, and I will turn things over to April to talk about what we've been up to with culinary.
Thanks. Give me a sec to share. Mm-mm-mm. There we go. All right. We'll kick it off with a couple of things that people sent me throughout the month. April Wyman was in Switzerland and shared a couple of fun photos. Morels and more reasonably priced at a Swiss grocery store. Lucky them. And also this amazing looking homemade pasta with fresh black truffles and truffle butter. So she was eating well there. Thanks for sharing that. And Daniel Berman made this amazing looking chicken of the woods pot pie and shared that as well. I guess it is a recipe, a variation on the chicken pot pie recipe from Christine Ha, who I'm familiar with, but seems like an amazing cookbook. That would be a great addition for a future potluck or cooking competition. Just saying, Daniel. And then just yesterday after Sequinoda, I'm assuming, but maybe not, that she found this there, or maybe just remembered to send it. The Tylopilus baticeps, which I have never tried, that she just prepared very simply with olive oil and garlic, and so it had a great toasted marshmallow texture, crispy outside and pillowy soft inside, which sounds amazing. And then I've got a few slides just to showcase what we did with culinary at Sequinoda, including a great workshop that Amar Shah had led and I contributed to as well. People learned to make his famous balsamic, I think now famous balsamic marinated mushrooms, mushroom jerky, and then lobster mushroom rolls that we shared with the whole camp. And then I had a mushroom pate. Got pictures of these on the next slide. That middle one's the pate. And so right there, the lobster mushroom rolls. And on the left there was the oyster jerky. And then culminated with our second annual mushroom banquet that Chef Julian Fortu again put together for us. He brought 150. The last year there was some feedback that like mushrooms got a little lost in some of the dishes, and so he took that feedback to heart and brought 150 pounds and was I think he brought 13 species and it was 14 with the chicken of the woods that someone brought him. So many folks pitched in to help clean and chop and do other prep. And then we ended up having truffle flatbread, mushroom phyllo. Actually, let me go to the next slide where you can see some of this. Oh, let me stay there where I've got my notes. A mushroom taco bar with homemade tortillas, which I just had some leftovers scarfed before this meeting. Mushroom ceviche, and then again his candied chanterelle candy cap cheesecake. And there are a couple of photos there. And that's all I have this month. As Elizabeth said, we'll have the mushroom beer making on October 27th. That will again be at Waverly, so look out for an invite for that. Be limited to 25 people, but as always, if you go to register and it's full, do get on the wait list because often people do cancel and spaces will open up. That's all, and thank you. Oh, and yeah, send your pics if you got them to culinary@mawdc.org as we start to get into fall mushroom season. I'm sure you guys will be finding and making some amazing things.
Awesome. Thanks so much, April. It's always fun to see what people are sending in. Um, Annie, you are up next talking about all the mushrooms.
Awesome. Thank you so much, April and Elizabeth. I had a great time at Sequanota. It was just fantastic. And I am really excited for next year. I already started thinking about potential songs for the talent show, which was definitely a highlight. Let's see. I just want to just have this going. Oops, nope. Sorry. There we go. Slides look okay? Good. Okay. So this is my virtual ID table. This is actually a picture of somebody at NASA. It's a NASA connection. Someone at NASA doing a virtual reality thing in the 90s. I just thought it was a funny picture. So luckily we have real mushrooms, but we're IDing them virtually. So as Elizabeth mentioned, we did a special project in iNaturalist for Sequanota, which we've actually done every year for the past several years. So if you go into iNaturalist and under the search bar, you just type in Sequanota and it should automatically pop up Sequanota 4A projects from like, there's definitely 2022, 2023, 2024. There may even be more than that. And if you click on the 2024 project, it will get you to this front page. If you want to join the project, you can click right here. Right now, it would say leave because I've already joined, but if you wanted to join, it would say join. And as you can see, we have 53 people in the project, which is awesome. I would say because what did we have? Like a little over 100 people at Sequanota?
Yep.
So that means we're up to like almost 50% of people who are using iNat, which I mean, I think that's, I can't remember what it was last year, but I would say, you know, the uptake is good. I'd love to see it be 100 next year. Maybe everyone can get on, but still, I think that's pretty good. And a ton of observations. So down here, you can see, I'm sorry in advance, my son is playing Minecraft and very loud in the background. I hope you guys aren't able to hear it. Anyway, about 1,296 observations. Now, per iNat species count, we have 342 species. Per the table count that Mitch sent me, I think we were at 267. But I think that will probably go up. I think in the past couple of years, we've had a higher count than that. I mean, that's it's a lot of species, first of all, so we should feel great about 267. But one thing I think that is a trend that I'm noticing is we're more hesitant to put species names on mushrooms because our DNA work has revealed so many cryptic species. So it's kind of funny. It may initially appear that we're seeing fewer species, but I think actually we're starting to just put the genus name on some things that we're not sure about. And that brings us to the most incredible number, and that is 200 samples were taken for DNA sequencing, and Serenella and Megan led that with a lot of help from a lot of other people, Martin, John Plishke, many other people. I don't want to miss anybody, but it was a big group effort and excellent leadership. So 200 samples, that was the goal. It was achieved. Very cool. And they worked tirelessly to do that. And, people did also really important work going out and bringing in the samples. So just to go to the next page, you can see here, this is if you scroll down on the project a little bit, you can see the top contributors. So Karen Beal came in as the most observations and the most species. Fabulous job, Karen, my roommate. Powerful duo here in cabin 11. Things are great in cabin 11. Anyway, yeah, Karen just found really cool stuff and also was just identifying tirelessly and, not identifying things large and small and many, tiny ascomycetes and neat slime molds. Richard Jacob did some wonderful collections and found a ton of species, and John Harper and Serenella and Josh and all of these people, everyone did a great job. So even if you aren't in this top 5 list, you should feel great about your observations. In terms of the most observed species, it's actually a little bit different from last year. I think in previous years, we had a lot more Entoloma quadratum, which is it's a salmon colored entoloma, and that we didn't see much of that. I think there was only one collection, but we did see a lot of Lexinum longicurvipes, which I think we see every year, but that so far is the top thing we saw. Tawny grisette, that's Amanita fulva. Saw a lot of that. Interesting, Cordyceps militaris, the scarlet caterpillar club, made it into our most observed species, which is really interesting because that's not usually something that's super common. And I think I don't know about if Gymnopus dryophilus is always on there, but I definitely know that the purple cortinarius is always on there. And Areobolides annexus also was all over the place. So really cool stuff. And I just wanted to do a close-up on Lexinum longicurvipes because it was the most observed, and also because I noticed that a lot of people were either not knowing what it was, it was coming in with just like fungi, or people were there were some misidentifications too. So I thought this is a great learning opportunity, especially because it's a really nice mushroom. It's like a great edible, a nice bolete. It's pretty. So it seemed like a good one for us to learn about. So these are different pictures of Lexinum longicurvipes. And, you can see, does anyone want to, I guess we have people muted. If people want to write in the chat anything that they notice from these pictures, like any features that really jump out at you, go ahead and do that. And I'm not actually seeing the chat for some reason. Can anyone tell me what's coming up in the chat?
Yeah, slimy cap, reticulation, no gills, curved stipe.
Awesome. Yeah. That's great. So the first one is really important. The slimy cap, I'm really glad that is at the top of the list. No gills, yes. So, great. That's great. People are noticing this is one of the boletes, the fleshy pored mushrooms. So we have polypores, which also have pores instead of gills. That's their fertile surface. But polypores do not have this like kind of fleshy pillow-like quality in their cap. And so boletes have that. And boletes also have, you can't see it in these pictures, but they have the ability to really easily separate the layer of tubes, which is the kind of machinery that produces spores, from the rest of the cap tissue. And polypores, that also can't be done generally. So someone said, did someone say reticulation or netting? Actually, that'll be a point for us to cover because this is not reticulation or netting. So I'm going to talk about what it is in a minute, but I'm really glad someone brought that up because, we can just clearly see that the stipe or the stem has ornamentation, meaning there's like decorations going on in there. And that's a key part of IDing boletes, as anyone who was in Arlene and Alan Bassett's fabulous bolete workshop at Sequanota learned. For their books, a big starting point for which keys to use is what is the ornamentation on the stipe. So is it smooth? Is there netting? Are there other things? So I'm going to talk about that a little bit.
Cindy also pointed out that the stem is sort of proportionately longer than you see in a lot of other boletes.
That's great. So I'm really glad that we have the length of the stem, and we also have the curve of the stem, and that is, drum roll, where we get the name Longicurvipes, long curved stem. So that is really good noticing. Was there anything else, or is that pretty much it? I mean, that's a very good description.
A couple people have chimed in with the word scabrous for the stem.
Oh yeah, boy. I love to hear it. That's really good. So we'll talk about that. And there's also just a lot of copious white mycelium. I don't know how, a lot of mushrooms can have that, but I'm just noticing that right in front of me in the picture. But you can notice that the mycelium is clearly in the oak leaf litter, and that gives you a little hint about who this guy is growing with. So here's a description of Lexinum longicurvipes from Michael Kuo's website, mushroomexpert.com. And you can see the ecology of this. Has a mycorrhizal, or people say ectomycorrhizal, relationship with oak trees, and growing alone or gregariously, widely distributed. So I would say that fits here. Like I saw it pretty much almost on every different foray that we went on, which means a lot of different kind of microenvironments. And I see it here at home a lot as well. The cap is not particularly large, 2.5 to 5 centimeters. Convex, that just refers to the kind of fact that it's very cappy. And sticky is key. So another word for that is like viscid, when fresh, but when it's older, or if it's very dry, it could just be smooth. But definitely when it's young, it will be sticky. And when it's older and dry, you might see things stuck to it. So that can be a key to tell you that something was viscid, if there's leaves and stuff that are just stuck to the cap. A little bit wrinkled, orangish, brownish, goldenish, and the pore surface, dull yellow. The pores are, not bruising. So we know a lot of boletes bruise blue. These ones don't. They're pretty small, and it says angular. I mean, I think they're like pretty round, but maybe a little bit angular. Long stem, that's clear. Scabers, curved. Scabers is an important word, and we'll talk about what that is. Sometimes becoming nearly reticulate. So I don't want to say the person's wrong who said reticulate, but it's almost like a kind of, it's not really a real reticulation the way that, for example, Boletus edulis or Tylopolis phelleus would be just pure nets. This would be like, there are all these scabers and they almost end up looking like net shaped, but they're still, if you like really peer in at them, they're these like individual kind of little pokey things. Basal mycelium white, we saw that for sure. And I didn't show any pictures of the flesh, but I said the pores didn't turn blue, but actually if you bisect the entire mushroom, the flesh at the base might be a little blue. So that can be a tell also. There's a big chemical reaction that I know we've done in the past at Sequinoda, which is you put potassium hydroxide, a little KOH on the cap, and it just flashes bright red, very beautiful. So that's cool. And the spore print is olive brown. So I noticed that many people were calling this Sewillis, and I totally understand why. It is a very honest mistake to make, because Sewillis are slippery jacks or slippery jills. They are often viscid. Sometimes they're more than viscid. Sometimes they're just like glutinous or frankly slimy. Some are not, but that's just a fairly common feature of Sewillis. So I think when a lot of people were seeing the kind of tackiness, goopiness of the cap here, they were thinking Sewillis. But there are some important distinguishing features. So first of all, a lot of Sewillis have either a ring or veil, although not all do, but Lexinum longicurvipes doesn't. A huge key is the stipe ornamentation. So scabers in the Lexinum species, that's true of almost all Lexinum species, I guess all Lexinum species. Whereas the Sewillis species, if they have stipe ornamentation, will have what we call glandular dots or resinous dots. And I'll get into the details of how those differ. huge thing is environment. So Lexinum longicurvipes, oak, and Sewillis almost exclusively with conifers. The only exception that I know to that is Sewillis subaureus, which is a kind of boreal species that I think grows in the northern parts of the Midwest. It grows with aspen, quaking aspen, I think, but that's the only exception I know. Otherwise, Sewillis conifers. The pores of the Lexinum longicurvipes are smaller. Sewillis pores can be really large. They can be very stretched out and angular, and even sometimes they can be like radially arranged, getting into what we call boletinoid, where they're so stretched out that they almost start to just look like long wannabe gills. So anyway, the spore print for Lexinum longicurvipes is olive brown, whereas Sewillis are going to have a more cinnamon kind of reddish brown spore print. And these are a couple pictures I've taken, and I just thought it would be helpful to show a close-up. So the Lexinum stem, you can see there's clearly like a 3D element of this ornamentation, these little tiny pokey dots, and they're called scabers. Whereas with the Sewillis salmonicolor, which also there's Sewillis coternatus or coternatus. It looks very similar, and it's actually unclear whether these are the same species, but I'll just call this Sewillis salmonicolor because it is also the older name. You can see that first of all, this Sewillis has a big veil remnant or ring that looks like super goopy. You can also see there is ornamentation on the stipe, but it's these kind of dots. And I don't know if you can fully tell, but I believe you can. The dots are not raised. They're flat. They're like, if you had your eyes closed, you would not be able to feel those. Whereas here on the Lexinum, you would feel that it's rough. They actually even call Lexinum rough stocks. Whereas the Sewillis, yes, it's not in relief. So I just went into more details here about bolete stipe ornamentation. And scabers are made up of cystidia. Cystidia are sterile cells. That means they don't make spores, and they do other helper things for mushrooms. And for scabers, they form these pointy scales. The little pointy scales typically start out white, but they can grow darker with age, or more colorful. Usually in a lot of boletes, they can be, more brown or even can look black, like in the Lexinum scabrum group, which is the birch bolete group. But they can also look kind of reddish, which is how I think they look for Lexinum longicurvipes. They can leave a kind of bruleed or scorched looking stipe. Just to show you again here, it almost looks like, I think Arlene Bassett or Alan Bassett said it looks like someone took a lighter to the stipe and just kind of scorched it, which is, I think, a great way of describing it. If you touch these scabers, they are not going to stain your fingers colors. In contrast, the Sewillis has glandular or resinous dots, also little clusters of cystidia or sterile cells, but these ones make pigments. And they start off as sort of little kind of pinpoint dots, but they can merge to look like smears or patches. They feel smooth to the touch, meaning they're not raised and pointy the way that the scabers are. They can also feel tacky to the touch because they're glandular, they're making some kind of pigment, and they can stain your skin when you touch them. And the color of them also may change with time. Oh yeah, so here's the same picture, but here I said scabers and glandular or resinous dots. So that's what I wanted to tell you about Lexinum longicurvipes. It's a bit of a deep dive, but I now know that everyone who is here would be able to identify it and is not going to mistake it for a Sewillis. The only other mushroom that I really wanted to show, I think we'll do another thing where we show a broader view of Sequinota, but I think it's better to wait till we have kind of more information, starting to get some DNA information, more sequence counts. But I saw this from Martin, and I thought it was really, really cool, so I wanted to show it. When I first saw the pictures on iNat of this, I thought, it looks really funnel shaped, and I thought like, oh, it's like, is it a clitosoby, one of the funnels? But it was a name that I did not recognize. And that always throws me for a loop because, I spend a lot of time trolling the seas of INAT and Facebook and reading all these things. So when there's a genus that I have never seen before, I feel a little excited and scared. And that's how I felt when I saw Lulesia. But the name Mundula, that actually was a little familiar to me. And I'm like thinking, I know this mushroom, like I've seen this before, especially with these kind of concentric ring zone kind of stuff on the cap. I'm like, that looks familiar. I'm like, what is this? Who is this? I don't know. So I had to Google it. And I figured out that this was what we previously were calling Clitosella mundula. And there were other names for it too that I'll show you. We don't have to go into too many details, but yeah, this is an interesting mushroom because it really looks a lot like a funnel or a clitosobe. But in fact, you can see it has kind of pinkish hues at maturity, and the spore print is pinkish or dirty brownish pink, which is unique. It also has a strongly farinaceous odor. Farinaceous, people describe in all different ways. I think farina means flour in Latin. So people say it smells like flour or tastes like flour. Flour, not the plant, not like a beautiful rose, but like actually flour. Some people say it tastes like or smells like cucumbers or melons. It's like hard to put it once you've smelled something that's like allegedly farinaceous, I think you'll get the vibe of it, but it's so subjective. Anyway, but the pink spores is a kind of giveaway for where this sits. It is not a clitosobe. So I was thinking, well, what happened? Why was this Clitosella moved? When was it moved? How did I miss this Lulesia? I was upset. What happened? Why didn't anyone tell me? And I found this paper, Alfredo Vizzini, that he's published a lot of stuff that's really interesting and cool. And I was just reading, he had a huge paper showing like a lot of different species that were either previously called Clitosella or Clitosobe or other things that were moved to Lulesia. But I also noticed this really funny thing here in the paper. I guess it's not funny, but I was looking for my mushroom for Mundulus, right? And I couldn't find it in his paper. And I'm like, why didn't the paper specifically address that? And then I did a search for Mundula, and I saw Mundulus, and I saw they said Agaricus mundulus, blah, 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 were recently combined in Lulesia via the Index Fungorum e-publishing by Baroni et al. 2023 in a non-ethical predatory way, even though we agreed on joint work. So drama, mycology drama, people. That was like interesting. Would love to know the backstory on that. Anyway, there's a famous quote you've probably all heard: a toadstool by any other name would smell as sweet, by William Shroomspear. And, it doesn't matter what you call it. You can call it Lulesia or Clitosella or Clitosobe, whatever you want, but it's the same mushroom. It's cool. These are all the names that this mushroom has gone by. It is like clearly a lot. Yeah, Clitopolis, I also think that was one that I was aware of. So whatever. This is the phylogeny that just shows pink spores. I mentioned pinkish brownish spores. That's the giveaway for what kind of mushroom this is. It is related to the Entoloma species. So up here, the first branch is Infundibulosobe, which is, one of the funnels, but that branches off way earlier. And then here you have at the top Entoloma sensulato, and these are all kind of Entoloma cousins: Rhodophana, Rhodosobe, Clitopilopsis, Clitopilus, and then here the new Lulesia, formerly Clitosella. You don't have to understand a phylogeny, but the main thing is, it just shows you how closely related different species are to each other. So you can group them into clusters or clades, and they also give you a sense of when they diverged from one another in time. So, like way back in time, Infundibulosobe diverged from this whole group of pink spored Entoloma wannabes. I think that's it.